Welcome to our deep dive into sequencing technologies. In this video, we'll explore the fascinating world of DNA sequencing, a field that has revolutionized biology and medicine. Sequencing allows us to read the genetic code, unlocking secrets about life, health, and disease. From the earliest methods to today's cutting-edge tools, sequencing technologies have evolved rapidly, making genetic analysis faster, cheaper, and more accurate. Whether you're a student, a researcher, or just curious about science, this journey will help you understand how we decode the language of life. Let's begin by looking at what sequencing is and why it matters so much in modern science. DNA sequencing is the process of determining the exact order of nucleotides within a DNA molecule. Think of it as reading the instruction manual for life, letter by letter. This technology allows scientists to identify genes, understand genetic diseases, and even trace the evolution of species. Over the years, sequencing has become a cornerstone of research in genetics, medicine, and biotechnology. By knowing the sequence, we can develop targeted therapies, improve diagnostics, and gain insights into how organisms function at the most fundamental level. The journey of sequencing began in the 1970s with the development of the first DNA sequencing methods. These early techniques, such as the Sanger method, were groundbreaking for their time. Although labor-intensive and slow by today's standards, they paved the way for future innovations. Scientists would manually read DNA sequences, often working with radioactive materials and gels. Despite the challenges, these methods allowed the first genomes to be decoded, setting the stage for the genetic revolution that followed. Sanger sequencing, developed by Frederick Sanger, became the gold standard for DNA sequencing for decades. This method uses special chemicals to terminate DNA synthesis at specific points, creating fragments that can be separated and read. Sanger sequencing was used to decode the first human genome and is still used today for smaller-scale projects. Its accuracy and reliability made it a favorite among researchers, even as newer, faster technologies emerged. While early sequencing methods were revolutionary, they had significant limitations. The processes were slow, expensive, and required large amounts of DNA. Reading even a small genome could take months or years. These challenges motivated scientists to search for faster, more efficient ways to sequence DNA. The need for speed and scalability drove the development of new technologies that could handle larger projects and deliver results more quickly. Introduction of next-generation sequencing, NGS, marked a turning point in genomics. NGS technologies can sequence millions of DNA fragments simultaneously, dramatically increasing speed and reducing costs. This leap forward made it possible to sequence entire genomes in days rather than years. NGS opened new doors for research, clinical diagnostics, and personalized medicine, making genetic information more accessible than ever before. Next-generation sequencing involves breaking DNA into small fragments, attaching adapters, and amplifying them. These fragments are then sequenced in parallel, generating massive amounts of data. Advanced software assembles these short reads into complete sequences. This process allows for high-throughput analysis, making it ideal for large-scale projects like population genetics, cancer research, and infectious disease tracking. Illumina is one of the leading companies in next-generation sequencing. Their platforms use a method called sequencing by synthesis, where fluorescently labeled nucleotides are incorporated into DNA strands and detected by cameras. Illumina's technology is known for its accuracy, scalability, and cost-effectiveness, making it a popular choice for research and clinical applications worldwide. Torrent is another innovative sequencing platform that detects DNA sequences by measuring changes in pH as nucleotides are incorporated. This approach eliminates the need for fluorescent labels, making the process faster and more straightforward. Torrent technology is especially useful for targeted sequencing in clinical diagnostics, offering a flexible and efficient alternative to traditional methods. Single-molecule sequencing technologies, such as those developed by Pacific Biosciences and Oxford Nanopore, read DNA strands directly without the need for amplification. This allows for longer read lengths and the detection of complex genetic variations. Single-molecule approaches are transforming genomics by providing more detailed and accurate information, especially for challenging regions of the genome. Pacific Biosciences, single-molecule real-time (SMRT) sequencing technology captures DNA synthesis in real time. By observing the activity of a single DNA polymerase, SMRT sequencing can generate extremely long reads, which are valuable for assembling complex genomes and detecting structural variations. This technology is widely used in research that requires high accuracy and long-range information. Oxford Nanopore's technology uses tiny protein pores embedded in membranes to read DNA sequences as they pass through. Changes in electrical current reveal the sequence of bases. This portable and scalable technology enables sequencing in the field, making it ideal for rapid outbreak response and environmental monitoring. Nanopore sequencing is opening up new possibilities for real-time, on-site genetic analysis. Generation sequencing refers to technologies that read single molecules of DNA or RNA without amplification. These methods offer longer reads, faster turnaround, and the ability to detect epigenetic modifications. Third-generation platforms are pushing the boundaries of genomics, enabling new discoveries in areas like cancer, rare diseases, and evolutionary biology. Sequencing technologies have transformed medicine by enabling personalized treatments, early disease detection, and better understanding of genetic disorders. Doctors can now use genetic information to tailor therapies to individual patients, improving outcomes and reducing side effects. Sequencing is also used to diagnose rare diseases, track infectious outbreaks, and guide cancer treatment. In cancer research, sequencing technologies are used to identify mutations that drive tumor growth. By analyzing the genetic makeup of cancer cells, scientists can develop targeted therapies that attack specific vulnerabilities. Sequencing also helps monitor treatment response and detect relapse, making it a powerful tool in the fight against cancer. Sequencing plays a crucial role in tracking infectious diseases. By sequencing the genomes of viruses and bacteria, scientists can monitor outbreaks, identify new variants, and track the spread of pathogens. This information is vital for public health responses, vaccine development, and controlling epidemics. For patients with rare genetic diseases, sequencing can provide answers that were once out of reach. By analyzing a person's entire genome, doctors can pinpoint the genetic cause of mysterious symptoms. This leads to faster diagnoses, better treatment options, and hope for families affected by rare conditions. Sequencing technologies are used in population genomics to study genetic diversity across large groups of people. By comparing genomes, scientists can learn about human evolution, migration patterns, and disease susceptibility. These insights help improve public health strategies and deepen our understanding of human biology. In agriculture, sequencing helps breeders develop crops that are more productive, disease-resistant, and climate-resilient. By understanding the genetic makeup of plants and animals, scientists can accelerate breeding programs and improve food security. Sequencing is also used to monitor food safety and trace the origins of agricultural products. Sequencing technologies are used to study the genetic diversity of entire ecosystems. By analyzing DNA from soil, water, or air samples, scientists can monitor biodiversity, track invasive species, and assess ecosystem health. Environmental genomics provides valuable information for conservation and environmental management. Epigenetic sequencing technologies allow scientists to study chemical modifications to DNA that affect gene expression without changing the sequence itself. These modifications play a key role in development, aging, and disease. By mapping the epigenome, researchers can uncover new layers of genetic regulation and identify potential therapeutic targets. RNA sequencing, or RNA-seq, is used to analyze gene expression by sequencing RNA molecules. This technology helps scientists understand how genes are turned on or off in different cells and conditions. RNA-seq is widely used in research on development, disease, and response to treatments, providing a dynamic view of the genome in action. Genomics involves sequencing DNA from entire communities of organisms, such as microbes in the gut or soil. This approach reveals the diversity and function of microorganisms that can't be cultured in the lab. Metagenomics is transforming our understanding of microbiomes and their impact on health, agriculture, and the environment. Sequencing generates massive amounts of data that require powerful computational tools for analysis. Bioinformatics combines biology, computer science, and statistics to interpret sequencing results. This field is essential for assembling genomes, identifying mutations, and making sense of complex genetic information. With the explosion of sequencing data, storing and managing this information has become a major challenge. Researchers rely on advanced databases and cloud computing to handle petabytes of genetic data. Ensuring data security, privacy, and accessibility is critical for the future of genomics. The cost of sequencing has dropped dramatically over the past 2 decades. What once cost millions of dollars can now be done for a fraction of the price. This reduction has democratized access to sequencing, enabling more researchers and clinicians to use these powerful tools in their work. Modern sequencing platforms can process thousands of samples in parallel, delivering results in hours or days. This high throughput is essential for large-scale projects like population studies and disease surveillance. Advances in automation and robotics have further increased speed and efficiency. Accuracy is critical in sequencing, especially for clinical applications. Modern technologies have greatly reduced error rates, but challenges remain, particularly with long-read platforms. Ongoing improvements in chemistry and software are helping to ensure that sequencing results are reliable and reproducible. As sequencing becomes more widespread, ethical questions arise about privacy, consent, and data sharing. Who owns genetic information? How should it be used? Addressing these issues is essential to ensure that sequencing benefits society while respecting individual rights. Sequencing technologies are subject to regulations that ensure safety, accuracy, and ethical use. Governments and organizations set standards for clinical testing, data protection, and quality control. Staying compliant with these regulations is crucial for researchers, clinicians, and companies in the genomics field. Direct-to-consumer (DTC) sequencing services allow individuals to access their genetic information without a doctor's order. Companies like 23andMe and AncestryDNA offer insights into ancestry, health risks, and traits. While popular, these services raise questions about data privacy and interpretation of results. Sequencing is at the heart of personalized medicine, where treatments are tailored to an individual's genetic makeup. By understanding genetic differences, doctors can prescribe the most effective drugs and avoid those that may cause adverse reactions. This approach is transforming healthcare and improving patient outcomes. Pharmacogenomics uses sequencing to study how genes affect a person's response to drugs. This information helps doctors choose medications that are safe and effective for each patient. Pharmacogenomics is a key component of personalized medicine and is being integrated into clinical practice worldwide. Sequencing technologies are used in prenatal and newborn screening to detect genetic disorders early. This allows for timely interventions and better outcomes for affected children. As sequencing becomes more affordable, its use in routine screening is expected to grow. Sequencing is a powerful tool in forensic science, helping to solve crimes and identify victims. By analyzing DNA from crime scenes, forensic scientists can match suspects, exonerate the innocent, and provide closure for families. Sequencing has become a standard part of modern forensic investigations. Sequencing technologies are essential for gene editing, especially with tools like CRISPR. By reading and verifying genetic changes, scientists can ensure that edits are precise and effective. Sequencing also helps monitor off-target effects and improve the safety of gene editing therapies. In synthetic biology, sequencing is used to design and build new biological systems. By reading and writing DNA, scientists can create organisms with novel functions, such as bacteria that produce medicines or clean up pollution. Sequencing ensures that synthetic constructs are accurate and function as intended. The future of sequencing is bright, with ongoing advances in speed, accuracy, and affordability. New technologies are emerging that promise to make sequencing even more accessible and powerful. As we continue to unlock the secrets of the genome, sequencing will play an ever greater role in science, medicine, and society. Portable sequencing devices like Oxford Nanopore's MinION are making it possible to sequence DNA anywhere, from remote field sites to space stations. These compact tools are revolutionizing research by enabling real-time analysis in challenging environments. Portable sequencing is expanding the reach of genomics beyond the traditional laboratory. Artificial intelligence (AI) is playing a growing role in sequencing, helping to analyze data, identify patterns, and predict outcomes. AI-powered tools can process vast datasets quickly and accurately, accelerating discoveries and improving the interpretation of complex genetic information. Sequencing projects often involve collaboration between scientists, institutions, and countries. Global initiatives like the Human Genome Project and the Earth Biogenome Project are mapping the genetic diversity of life on Earth. These efforts rely on shared data, open communication, and teamwork to achieve their ambitious goals. As sequencing becomes more integral to science and medicine, education and training are essential. Universities and organizations offer courses and workshops to teach the latest techniques and data analysis skills. Building a skilled workforce ensures that we can fully realize the potential of sequencing technologies. Engaging the public is important for the responsible use of sequencing technologies. Outreach programs, science communication, and transparent policies help build trust and understanding. Involving communities in research ensures that the benefits of sequencing are shared widely and ethically. Despite incredible progress, challenges remain in sequencing technologies. Issues like data management, interpretation, and equitable access must be addressed. Continued innovation, investment, and collaboration will be key to overcoming these hurdles and maximizing the impact of sequencing. Sequencing technologies have transformed our understanding of life, health, and disease. From the first genomes to today's real-time portable platforms, the journey has been remarkable. As we look to the future, sequencing will continue to drive discoveries, improve medicine, and shape our world. Thank you for joining us on this exploration of sequencing technologies. Stay curious and keep exploring the wonders of science.